Dear students, welcome to our lecture number 9 of Zoo 301 Physiology. Now we will study the digestive glands. In last topic we have seen the gastric digestion, the gastric juices, and just started intestinal digestion. So in intestine, main digestive glands, which includes pancreas and liver. In liver, we have seen last time that gallbladder is there, which secrete bile juice, and in pancreas, the pancreatic juice and insulin. These two juices are secreted, or these two which controls the metabolism of food in intestine. Now, the pancreas is a dual gland. It is connected to the loop of duodenum, and for their secretion either directly or by the ducts. The pancreatic juice secrete pancreas secrete pancreatic juice, which is secreted by the pancreatic duct, whereas the endocrine secretion, that is insulin, is directly poured into the bloodstream, and this is the two ways by which the pancreas controls the metabolism. Therefore, it is also called as heterocrine or dual gland. Heterocrine or dual gland. If it secretes by duct, it is exocrine. If it secretes directly, called as endocrine. Therefore, exocrine and endocrine, both type of glands are there. So enzymes which are secreted by this pancreas are suspended in very alkaline pancreatic juices. Means the nature is alkaline and includes amylase, which we have already seen in mouth, which contains saliva as a ptyalin. This amylase is also present in intestine, and for breaking down starch into sugar, we have seen that the sugar is nothing but the maltose sugar. And lipase, it digests not only sugar but also lipase or lipid materials when activated by bile salts, and helps to break down the fat material. So they help to digest the carbohydrate as well as fat, in short. So the hormone insulin, you see here is the insulin which stimulate to control the blood sugar level, and produced by specialized cells called as the islets of Langerhans. The insulin is secreted by islets of Langerhans in pancreas and plays important role in controlling the blood sugar level in the blood. Next, the pancreatic juice is secreted by exocrine part, that we have already seen, but it is secreted from the acids, or we can say small lobules, in which the pancreatic juice is stored, and it secretes alkaline secretion, and its main contents are trypsin. Trypsin is made up of 223 type of amino acid molecules, and it can digest proteases and peptones. So we can see here a simple reaction: trypsinogen converted into trypsin in presence of an enzyme enterokinase. One simple thing to be noted here and keep in mind is that whenever we use ASE, it means that is generally in bioscience, it is an enzyme, because ASE is the term attached to the word, specially works as an enzyme. So in next we see that proteins, peptones, proteases, these all are digested into polypeptides and free amino acids in presence of trypsin. Second, pancreatic chymotrypsins. These are in the form of chymotrypsinogen and activated in presence of trypsin. So trypsin is again which activate the chymotrypsinogen or chymotrypsins also. So in next reaction, carboxypeptidases, as I told you previously that ASE are the enzymes. The enzymes which digest carbohydrates are called as carbohydrates, and the enzymes which digest carbohydrate as well as peptides called as carboxypeptidases. So these are in presence of trypsin converted into carboxypeptidases. So these are One more thing, that the enzymes themselves play a key role in the metabolism of food which is in the intestine. So polypeptides in presence of carboxypeptidase converted into lower polypeptides or free amino acids. Similarly, the amylase converts starch, dextrin and glycogen into the maltose sugar that we have previously studied in the oral digestion. Now, lipase or stearin are fat splitting enzymes of pancreatic juice. It splits fat and oils into glycerol, fatty acids, monoglycerides and diglycerides. So pancreatic juice also has following enzymes, that is phospholipase, generally called as lecithinase or lecithin kinase, and another is cholesterol esterase which catalyzes free cholesterols. Then nucleolytic enzymes are also digesting some nucleic materials like ribonuclease, RNase and deoxyribonuclease or DNase, generally called as, or dornase. This is another name to DNase enzyme. Then bile juice. Bile is a digestive juice secreted by liver cells and stored in a gallbladder. And from gallbladder it is secreted by bile duct into the duodenum. Like pancreatic duct, bile juice is also secreted by bile duct from the opposite side in the duodenum. The liver cells produce about 0.5 gram of bile salts daily. So this one is also given at the end of this slide. The liver cells produce 0.5 gram of bile salts. So bile salts are different kinds that we will see later on. The liver, which acts as a large reservoir and filter for blood, it occupies the upper right portion of the abdomen and has several important functions. So different functions carried out by liver are this: one, secretion of bile to the gallbladder, or also known as choleresis. Then carbohydrate, protein and fat metabolism, or we can say it metabolizes all food ingredients, because these are the three main food ingredients of the food. The storage of glycogen keeps ready for conversion into glucose when energy is required. Means after digestion, after metabolism, whatever the food is stored in the liver cells or body cells, whenever they requires metabolism and production of energy, then and then the activity is converted by these cells. Okay, so storage of vitamins and filtration of blood is another function. Then phagocytosis is very important function carried out by liver, which ingest worn out red and white blood cells as well as some bacteria. Means they prevent the body from the infection. They ingest the foreign bodies in simple way, or dead and decaying material in the body. The liver cells also produce 0.5 gram of bile salts daily. This is the amount or the proportion secreted by liver cells. So the gallbladder is the organ where the bile juice is stored temporarily and then secreted into the duodenum, and concentrates bile or concentration of bile salts responsible for the emulsification of fat materials, and we can say metabolized simply, and it makes it easy to break down the food material by the pancreatic juices. So it also, bile juice also helps the pancreatic juice, and both collaboratively digest very well the food material in the intestine. So after that the food passes into the large intestine, and in a large intestine the average 1.5 meter long, and it comprises, large intestine comprises cecum, appendix, colon and rectum. These four organs also take part in digestion. Mainly they digest the water, minerals and other required elements or salts mostly. After food passes into the cecum, a reflex action in response to the pressure causes the contraction of the ileocholic valve, means ileum and colon. In between these two, there is a valve called as ileocholic valve that prevent any food returning into the ileum. Means they just pass the food into the colon, never allows to back into the ileum. Therefore, the valve is here. Here, most of the water is absorbed. As I told you that most important function in a large intestine which is carried out is the absorption of water, and much of which was not ingested. In the food, everything is not digested or everything is not there, but much of which is not ingested but secreted by digestive glands further up the digestive tract. The colon is divided into two lobes or stands, we can say, ascending as well as descending. In between that, the material is traversed through the tube, and before reaching the anal canal or near the anus, where the indigested food material is stored and expelled or ejected or thrown out from the body. And in this way, this digestion is here completed. But one more thing to be known about the intestinal juices which are secreted in duodenal glands. These are the unicellular glands which are located in the digestive system, or we can say intestinal wall. These are the Brunner's glands, which are found in the submucosae of the duodenum and produce alkaline secretion containing mucus mostly and helps to digest the food materials. Then the intestinal glands also present in the randomly scattered wall of the intestine and or in the crypts of liver cone. These are nothing but the contours, or we can say folds of mucus on which these crypts of liver cone are located, or we can say villi. Villi are also the microfolds which take part mostly in the absorption of food material after digestion. So, crypts of liver cone and the intestinal glands, these cells secrete large number of enzymes, and collectively, all the enzymes are collectively called as succus entericus, which are poured at a villi. The enzymes in intestinal juice includes peptidases, enterokinases, carbohydrates, and nucleases that we have already seen in previous slide. So, the enzymatic activities includes first peptidases found in the intestinal juice and hydrolyzes the peptides into amino acids, and these are of also two types. One is amino polypeptidases and second is dipeptidases and tripeptidases. This is the group of di and tripeptide groups. So what are the enzymes secreted in intestinal wall? How do they work as a metabolism? So before entering into the large intestine or at the end of large intestine, so these are the enterokinases, carbohydrates, sucrase, maltase, lactase, and nucleolytic enzymes. So they digest or activates on trypsinogen and converts into active trypsin. So carbohydrates include disaccharide splitting enzymes like sucrose, maltose, and lactose, and provide the energy whenever required to the body. And sucrase is an enzyme also known as invertase enzyme. It breaks the sucrose into the glucose and fructose molecule that we have seen in the last slide, or here we can see this is the simple slide that sucrose is split down by the sucrase enzyme. ASE means this is enzyme which splits them into the glucose and one molecule of glucose and one molecule of fructose. The maltase we have already seen. The maltose sugar converts into the molecules of glucose, and maltose is converted by maltose is converted by amylase from starch into the maltose sugar. So lactase, so this is the inverse reaction, or we can say reverse reaction. Food material from the digestive tract is digested and converted into the glycogen in the body. Glycogen is converted into the energy, and this is the another metabolism. This is the first metabolism in the tract, this is the second metabolism. Therefore we are again studying it in details. The lactase is the enzyme. Lactose is the sugar present in milk, and lactose is digested by lactase enzyme into molecules of the glucose and galactose. You can see here in this simple reaction, the lactose sugar is digested into glucose and galactose in presence of the enzyme lactase. Then nucleolytic enzymes, these enzymes hydrolyze nucleic acids only, DNA and RNA. Of them it includes nucleases. Nucleases specially hydrolyzes nucleic acids into nucleotides or mononucleotides. Already we know about the nucleotides and nucleosides which are present in DNA. Then phosphatases, this breaks the nucleotide into the nucleoside and phosphoric acid. So these are the two molecules. Then nucleosidases also cleaves means breakdown, cleaves nucleosides into free nitrogen bases and pentose sugar. Pentose is 5 group containing. Then in digestive tract, the digestive enzymes play various digestive metabolic activities. Some of these summarized here. Dipeptides converted into amino acids in presence of dipeptidases, or we can say dipeptidases acts on dipeptides and converts into amino acids. Simply, likewise, maltose is converted into glucose and glucose, one glucose, two molecules in presence of the enzyme maltase. Lactose is converted into glucose and galactose in presence of lactase enzyme. That we have seen in the recent activity, the previous slide. Then sucrose is another sugar which converts glucose and fructose molecule in presence of the enzyme sucrase. And nucleotides, we have seen that nucleotides are converted into nucleosides in presence of nucleotidases, and nucleosides are converted into sugars and base pairs or bases in presence of nucleosidases. So this nucleotide, nucleoside. So these are the different words. And di and mono. Di means double and mono means single. Di and monoglycerides are converted into fatty acids and glycerol. This is a simple digestion of fat in presence of the enzyme lipase. ASE. Enzymes which digest lipids are called as lipases or lipids. In this way, we have studied the activities of digestion in intestine. Now this is the summary chart giving the nature of digestive enzymes and their action. So these are the enzymes which we have already studied. Then these are the product, or we can say produced by the glands or organs. These are the sites where the digestive juice secretes, it's a secretion, and these are the different pH levels, either acidic, basic or neutral. So in salivary amylase, this is the enzyme which is secreted by salivary glands and poured its secretion into the mouth, and these are neutral. Whereas the pancreatic amylase, maltase, then trypsin, then peptidases, nucleases, nucleosidases and lipase, all these are basic digestive juices, means they have their pH more than 7, are the basic and secrete to the respective organs as described here in the chart or table. And only this pepsin is a gastric enzyme which is secreted by the gastric glands in the stomach and pour its secretion already in the stomach. And this is only acidic. We can say that the parietal cells which secrete hydrochloric acid we have seen in a stomach digestion, or whatever the cells which are responsible for the secretion of HCl, hydrochloric acid, are the acidic in nature, remaining are basic. So this is a very simple chart or table which gives the idea of the enzymes, their respective organs, and acidic or basic or neutral level. So at last we will see how the digestion of carbohydrates, proteins and fats are taken place or takes place. We have already seen everything in previous slides, but this is the summary chart in which the starch is converted into sugar. Maltose we have seen in presence of amylase, and that all sugars, not only maltose but all sugars are stored in the form of glycogen in the body. And whenever they require energy, then that glycogen are also split down into disaccharide and monosaccharides. So disaccharides are maltose, sucrose and lactose, and they are converted into glucose, fructose and galactose respectively. We have seen already in the previous chemical chains or metabolic chains. So maltose is converted into glucose molecules in presence of maltase. Sucrose is converted into glucose and fructose molecule in presence of sucrase enzyme, and lactose is converted into glucose and galactose molecule in presence of the enzyme lactase. This is very simple carbohydrate digestion. Now we will see the protein digestion. So digestion of proteins is also takes place. These are the reactions taken from the website. Trypsinogen converted into trypsin in presence of enteropeptidases or trypsin. This also itself work of conversion of metabolic activity. Then chymotrypsinogen converts into chymotrypsin in presence of trypsin. This is the reversible reaction. Pro-carboxypeptidases converts into carboxypeptidase A, carboxypeptidase B and aminopeptidases. Here this is also a group of peptidases and in presence of the trypsin. So trypsin is the hero here, or main element which is responsible for the conversion. So pro-elastase converts to elastase in presence of trypsin. So pro-polypase converts into polypase in presence of trypsin. So these are the another protein digesting materials or enzymes convert these activities for the completion of metabolism. And now the digestion of fats or lipids. We have already seen in stomach they partially digest monoglycerides, and next small intestine in which fat, emulsified fat, diglycerides, monoglycerides, emulsified fat, these all elements are digested in a small intestine. As we have already seen that all carbohydrates are digested into sugars, then all proteins are digested into amino acids, and likewise all fat materials are digested into the fatty acid and glycerols. That may be the monoglyceride, di or tri molecules, whatever the molecule number is there too. So monoglyceride are mostly from the diglycerides and splits into fatty acid and monoglyceride molecules. And everywhere the pancreatic lipase is responsible, except intestinal lipase, which is converting emulsified into monoglyceride and fatty acids. So this is a simple way which converts the fat into emulsified fat in presence of the bile and pancreatic juice that we have already seen. So this is about the digestive juices which are converting the food materials into the simple molecules, and it becomes easy for the absorption to the intestinal wall. So this is about the pancreatic juice, bile juice and intestinal unicellular gland cells which secrete the juices. And in this way the digestion of intestine is completed here. So next time we will see lecture number 10. The further topic is of absorption, how the food material is absorbed in a intestine. So till then, see you.